Good day, ladies and gentlemen. With this video, I'm going to show you how to use a volume meter to measure the rate of respiration of germinating pea seeds, and you can use then also this experiment to measure the rate of photosynthesis. Now, when we measure the rate of anything, we must remember that that takes time over a certain time period. And when we measure the rate of photosynthesis or respiration, we have to measure something that is being used or something that is being produced. So if we look at respiration, what is being used by respiration? During respiration, seeds or plants or animals will use the air that contains oxygen and breathe that in, and then also it will use glucose or any starch or something that is being stored for energy, and that is then broken down to produce energy, carbon dioxide and water vapor. So we can either measure the amount of oxygen being used or the amount of glucose that's being used, or we can measure the amount of carbon dioxide being produced or water vapor being produced, or the amount of energy or ATP being produced. Now for today, we are going to measure the amount of oxygen being used. And for that, we are going to use our volumeter. Every good scientist will always have a control. So the first thing that we are going to set up is our control, and we call that a thermobarometer. We are going to take this volumeter and we are going to keep it empty because we want to measure everything that in the environment can add on to or subtract from our experiment. And that is why it's called a thermobarometer, to measure changes in temperature and also changes in air pressure. How are we going to do that? The thermobarometer is an empty tube with a lid. Now, when we fit the lid, we have to take care to fit it very tightly, and therefore we have to press down and give it a twist. You will see there is a little plunger and syringe here that we can use, and then we have this side arm that is specifically made. It is a capillary tube, which means that if we fit the lid correctly, it will take some of this red fluid up by itself because it is a capillary tube. And there you can see the red fluid. And then we are going to put it down. This is our control. It might need some time to equilibrate. If there's any changes in temperature or in the air pressure, this air bubble or this red bubble, which is just water and food coloring, will start moving. Now we are going to set up our experiment with our pea seeds. Now we use pea seeds because they will not photosynthesize, so we won't have a production of oxygen. We will only be able to measure the usage of oxygen. And then we are going to put here on top of it some cotton wool, and you fill this pea seeds only about 3/4 of the way, and then we put the cotton wool on top. And the cotton wool is just so that we can separate the soda lime that we are going to layer on top of these seeds. Now, what is the function of the soda lime? The function of the soda lime is to absorb all the carbon dioxide that is being produced. We're going to use a spoon, and then we are going to layer it on top. Why do we want to use, or why do we want it to absorb all that carbon dioxide? Because during respiration, for every oxygen that is being used, a carbon dioxide have to be produced with some water vapor. Now we are going to set up our experiment. And when we set up the experiment, it is very important that we make sure that these tubes sticking out at the bottom do not touch our pea seeds and the soda lime. And then we are going to use our plunger, and we are going to take in a droplet. Just as before, we are going to put it down because we don't want to transport too much of our heat from our body onto our tube so that we have movement due to the heat. And then this one would have been calibrated already, equalized, and we are going to take our marker pen and we are going to make a mark where the droplet is. Now, as soon as we see this droplet in the one with the experiment, the one with the pea seeds starts to move, then we start to time, because remember, we are going to determine the rate of respiration. And depending on how fast the movement is, we will make a mark every 30 seconds or every minute. And each of those will then count as a measurement. We will also look at the thermobarometer at the same time, because if there's changes due to temperature or air pressure, then we have to add it or we have to subtract it. If the droplet in the thermobarometer moves in the opposite direction, we have to add the distance moved by the droplet. If it moves in the same direction, we have to subtract it from the droplet moved and the distance moved from this droplet. So as soon as we saw movement, then what did we do? We start our timer, and you can use your cell phone timer, you can use your watch, and then every minute we made a mark. And you will see not every mark will be equally spaced, and that just depends on how the oxygen is being used. So what is happening? If we look at our experiment, we will see the pea seeds is germinating, so they need energy to grow, and they will be using all the air that is in this tube. Eventually, they will start using the air that is in this tube, and they will suck that air bubble or that red bubble down the tube. And the speed or the rate at which it sucks that bubble down the tube is how much oxygen is being used in a minute. This tube, that is our experimental control, we did not see any changes, which is also correct, because we don't expect any sudden changes in the temperature or in the air pressure while we are doing our experiment for such a short period of time. And therefore, we are just going to say that measurement is 0. Now, how do we take our measurements? Once you have done 5 measurements, what you do is you take your ruler, you use the millimeter side, and then you measure from the first mark up to the second mark. And you write that down. And your second measurement is from that second line to the third line. And you write that down, and then that measurement. Now you have to be careful, this setup is not very stable, so you will always have to pay attention of what is happening. So you measure from that mark to the next mark. Now we are going to use those measurements in class, and I will explain to you how we are going to calculate how much oxygen, volume of oxygen. Why do we say volume? Because it is in a tube, it is in a cylinder that is being used by these pea seeds per gram fresh weight. Why per gram fresh weight? Because if we have 10 seeds, they will take up a lot more oxygen than if we have 2 seeds. Now, instead of counting all those seeds, we will be weighing them, and we know how many grams of seeds we have.